Let us now look into the anatomical features of the stems. To take up the dicot stem, the anatomy of a dicot stem can be studied by taking very thin sections of Helianthus annuus stem, that is the stem of sunflower plant. When we prepare a slide and observe it under the light microscope, the most prominent thing that is visible is the outermost skin of the stem, what is called the epidermis. It is the protective layer. In fact, this epidermis of the dicot stem has a very thin layer of cuticle, and it has innumerable fine multicellular hairs called trichomes. If the stem is herbaceous, we may even see a few stomata at the epidermal layer. Next to the epidermis, in the dicot stem, we can see a few layers of thick-walled collenchymatous cells constituting hypodermis. It provides mechanical strength to the plant body, and this hypodermis, comprising of collenchyma, consists of all living cells which in herbaceous plants are capable of performing photosynthesis. Next to the hypodermis, we see the cortex. The cortical cells below the hypodermis are rounded, thin-walled parenchyma cells with very conspicuous intercellular spaces. The cells have a central vacuole with peripheral cytoplasm and small nucleus in each cell. The innermost layer of the cortex is called endodermis. When we look at the endodermis, we find that inside these cells there are starch grains, and hence sometimes endodermis is also referred to as starch sheath. Next to the endodermis, all the tissues in the center of a dicot stem constitute the stele. In the stele, the outermost area is the pericycle, and this pericycle is present on the inner side of the endodermis. The pericycle is represented by two types of tissues. Wherever the vascular bundles are present, the pericycle shows sclerenchyma, and between the vascular bundles, the pericycle has parenchyma. Next to the pericycle, in the dicot stem, we can observe vascular bundles which are arranged in the form of a broken ring. This very arrangement of the vascular bundles in the form of a broken ring is the unique feature of dicot stem. If we look at each vascular bundle, we see that the xylem and phloem are found in the same radius, one above the other, and hence the vascular bundles are conjoined and collateral. A close observation of each vascular bundle shows the presence of thin-walled meristematic tissue between the xylem and phloem, and this is what is called vascular cambium or intravesicular cambium. As the cambium is present, the vascular bundle is said to be open. If we look at the xylem shows endarc arrangement, where the protoxylem faces the pith and the metaxylem faces the periphery. So, in the stele of a dicot stem, we find the vascular bundles arranged characteristically in the form of a ring within the endodermis, and at the central portion of the stem we observe lot of loosely arranged living parenchyma cells forming a prominent pith. Between the vascular bundles, it appears as if the parenchyma cells of the pith radiate towards the endodermis. Such parenchyma cells between the vascular bundles constitute the medullary race. If you look at a monocot stem, here also we do find almost the same kinds of tissues, but the arrangement is entirely different. As an example of monocot stem, we can take the section of maize stem and observe under the light microscope. Here also we find an epidermis, which is the outermost layer, but in the epidermis, the multicellular epidermal hairs or trichomes are totally absent. Next to the epidermis, in the monocots, there is hypodermis, and this hypodermis is basically made up of sclerenchyma tissue. The sclerenchyma is present in 2 to 3 layers, and in the ground tissue present within the stem, we find lot of vascular bundles scattered. There is no specific arrangement, and all these vascular bundles could be seen haphazardly arranged in the ground tissue, which is made up of parenchyma basically. Each vascular bundle, when magnified, looks like a skull. In the vascular bundle, there are present 2 rounded broad metaxylem, a protoxylem facing the center, and a water canal next to the protoxylem. Above the xylem is present the phloem, and the protophlloem is slightly crushed. We do not find phloem parenchyma. The entire vascular bundle is surrounded by sclerenchyma tissue, forming what is known as the bundle sheath. The water containing cavity gives, along with the metaxylem and protoxylem, the characteristic skull-like appearance to the vascular bundles. The vascular bundles that are present at the peripheral part of the monocot stem are smaller in size when compared to those which are present in the middle portion or the central portion of the monocot stem. As in the monocot stem, the vascular bundles are scattered and are arranged haphazardly, it is not possible to undertake grafting work in monocot stems. So thus, in the monocot stem, the main mechanical tissue is a sclerenchyma, whereas in the dicots, it is collenchyma. Sclerenchyma, though they are dead, they are able to provide sufficient strength to the stem.